Hello, can you hear me? Yes, good. People, we have to discuss today paper number 21. Paper number 21. Please find the revision BioEve paper 21. BioEve 21. Uh, this is Wednesday, 8:30 p.m. Discussion, people, paper number 21. Right, so let's discuss this. Let's discuss this paper. And hope you have received the paper. You can hear me now, right? Can hear me, right? Hope you all have received the paper and you have it right now. So we'll go on discussing it quickly. Characteristic of water responsible for organism. Character of water responsible for an organism can release much heat energy with minimum loss of water. I'm taking second one as the answer, right? Second is the answer. I'll go and write in the answers. Yeah, otherwise it will trouble me. So first question answer is to how many questions are there? How many questions present in the paper? 23. Right? Write them. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. All right, we'll go with the answers. First question, what is the answer, people? Second one, hope it's clear for you, because to maintain minimum, with a minimum loss of water, to maintain temperature, it has to be high latent heat of energy. So we'll take second one as the answer. The structure responsible for making a brush border, people, brush border is microvilli. We found it one point, somewhere we discussed about brush border. Right? Brush border is microvilli, people. The answer is the first one for question number 2. Question number 3, people, mitotic division. In mitotic division, centrosome move towards the opposite poles of the cell at the first stage of the prophase. Centrosomes move towards the opposite poles at the first stage of this prophase. Movement of centrosomes happen due to lengthening of the aster. Wrong, neither. Aster is not lengthening. What is aster? The fibers which go and attach to the cell membrane. What do you call the fibers? The spindle fibers go and attach to the cell membrane is forming the aster. So it says that movement of the centrosome happen due to lengthening of the aster. No. The centrosomes are moving away from each other. People growing the spindle fibers, right, present between them. We can't agree with that, right? Cohesion between sister chromatids are remained during the metaphase. Cohesion between, that's correct, neither. Sister chromatids are held together, people, until it pass the metaphase, and then at the anaphase only we can see that they are going to separate. So third statement is okay with me. 2 identical daughter cells are formed at the end of the telophase. No, at the end of the telophase, 2 identical nuclei are going to form, not 2 identical daughter cells. Wrong, right? And then at the end of the anaphase, nuclear envelope is formed around chromosomes. No, telophase it happens, right, people? So if you take all these points, they have not asked you to find whether the correct or the wrong statement, but anyway, you can see 4 statements are wrong. 1 statement is correct. So ultimately, people, you can mark that. So my answer is the third one. For third question, my answer is third. Question number 6. Question number 4. The process shown by the equation C6H12O6 plus 6 oxygen, carbon dioxide, water, 30 ATP, right? We know 32 ATP produced by people. Most efficient cells, like in liver cell and heart muscle cells, the total number of net ATP production is 32. Say they say people 30. So it got to be, right? It got to be. So in the third question, why the first statement is wrong? The centrosome move towards the opposite poles at the first stage. It's not at the first page, neither. Sayuri, they have given it first, second, third, little later, right? It's given little later. So check that note. Check that note. It says it is prophase, mitosis or meiosis, mitotic division. Mitotic division, first one, chromatin fibers get condensed. Number two, nucleoli get disappeared. Number three, the formation of mitotic spindle. Number four, centrosomes move towards the opposite pole. So, it's given as a fourth point. Given as fourth point, all right? Not as a first point in prophase. Fourth point. So, there is some order there, right? Question number 4, the process shown in the equation, reduction and endogenic. No, this is always oxidation process, right? Formation of carbon dioxide water is oxidation, so you can't take that reduction. Now, you know people, formation of carbohydrate is a reduction. This is carbon dioxide and water is an oxidation, right? So, cell respiration is oxidation. Photosynthesis is a reducing process. Calvin cycle is a reducing process. Krebs cycle is an oxidative process, right people? So, reduction, no. Take place in liver cells, no. It got to be 32 for liver cell. Reduction and exergonic, no. Take place in brain cell, possible. Why? Other than heart and liver, any other cell, 30, right? According to our syllabus, right? Neither an oxidation, no. Bullshit, ne? So, fourth one is the answer. Am I? Fourth one is my answer for fourth question. Question number 5, which of the following statement is correct regarding electron flow of light reaction of photosynthesis? Which of the following statement is correct regarding the electron flow of light reaction of photosynthesis? The excitation of PS1 and PS2 embedded in the thylakoid membrane synthesizes ATP and NADH. The excitation of PS1 and PS2 embedded in the thylakoid membrane synthesizing ATP and NADP. NADH. Wrong, Nedalamai. Excitation of PS1 and PS2 embedded in the thylakoid membrane synthesizes ATP and NADH. Wrong. NADPH is correct. NADH is wrong. Alright? Electron, they are asking correct one. Huh? Electron release as a result of splitting of water may neutralize the excited P700. Electron release as a result of splitting of water immediately neutralizing PS1. Wrong. Splitting of water due to non-enzyme catalyzed reaction. Splitting of water due to non-enzyme catalyzed reaction produce oxygen, H plus ions and electrons. Splitting of water due to non-enzyme catalyzed reaction, wrong. This is said to be an enzyme catalyzed reaction. Wrong statement. Right? Dasun is asking second question, why can't we the cilia? Second question, microvilli. Structure possible for making of brushboard. Brushboard is not cilia. Brushboard is always microvilli, right? That's a rule. Right? Cilia can't make a brushboard. No further discussion on that. Microvilli form brush border. It's, I think, given somewhere. Given somewhere. I can't remember exactly now. We discussed it once somewhere. Can somebody remember where we talked about brush border? One point. Not in intestine. Somewhere we discussed, mention brush border. Right? That's microvilli. Right? Then another student asking, why could you please explain the answer for the fourth question? Fourth question. My answer is fourth one, take place in brain cells. 32 ATP produce, right, in heart muscle cell and liver cell. Any other cell, 30. Any other cell, 30. What's the problem? Right? Heart cells, liver cell, what you said, maximum ATP production, 32, no? Net ATP production is 32. So, any other cell, 30. So, I'm taking fourth. Right? What else? What is the alternative answer you have? Come on, tell me. What is the answer you have chosen? What's the answer you have chosen? Sayuri? What's the answer you have chosen? Third one. Reduction can't be reduction. How can it be reduction? It's an oxidation process. That's wrong. How can this be glucose to carbon dioxide? How can it be a reduction process? It's an oxidation process. Wrong. Answer is fourth. All right. Sir, doesn't heart muscle cells produce 28 ATP? No, 32 if you take. Now, I'm not talking about the oxidative phosphorylation. I'm not talking about the electron transport chain. I'm talking about the total ATP production. Total ATP production, how many ATP? Total ATP. How can it be 28? 32, no? Right? How can it be 28? 32. Right? How we calculate that? 10 NADH. How many ATP? 2.5. 25 ATP here. And then 2 ATP directly. Then 2 ATP from Krebs cycle. Then 2 ATP from aerobic respiration, glycolysis, 2 ATP, net ATP. And then 2 FADH2, that is 3 ATP. What's the total then? What's the total? 32. No? What's the problem? What is your problem? Right? Answers for question number 1, 2, 3 marked here. So don't ask them again. Right. Next question, people. So fourth question, my answer is fourth. Question number 5. Which of the following statement is correct regarding electron flow of light microscope, electron flow of light reaction of photosynthesis? The excited PS1, PS2 are embedded in thylakoid membrane, synthesize ATP and ADPH, wrong. Electron release as a result of splitting of water neutralizes P700, wrong. Splitting of water due to non-enzyme catalyzed reaction produces, no, enzyme catalyzed reaction, mentioned as enzyme catalyzed reaction, wrong. NADP plus reductase enzyme catalyzes the reduction of NADP plus. NADP plus reductase enzyme catalyzes the reduction of NADP plus, correct. NADP plus reduce into NADPH, correct statement. Cyclic electron flow synthesizes ATP, NADPH, wrong. Only ATP, wrong. So my answer for question number 5 is 4. Question number 6, which of the following statement is incorrect? Which of the following statement is incorrect? Which of the following statement is incorrect? Some of the organisms can produce carbon dioxide without using oxygen during respiration. Correct. They can produce carbon dioxide without using oxygen, anaerobic respiration, like in ethanol fermentation in some organisms. Correct. For some organisms, mitochondria is not essential to generate ATP in respiration. Correct. Bacteria, for example, they don't need the mitochondria. They don't have mitochondria. All right. Correct. Respiratory substrate completely decarboxylate in the matrix of mitochondria. Perfect. Completely decarboxylate. Yes, we agree with that. Final hydrogen acceptor in ethyl alcohol fermentation is ethanol. No. Final hydrogen acceptor in ethyl alcohol fermentation is acetaldehyde. Wrong statement. And then 2 ATP are used for initiative step prior to oxidation in glycolysis. Correct. Initially 2 ATP utilized. Correct. So my answer for sixth question is fourth one. Is fourth. Hardalamai. Question number 7. Question number 7. Which of the following statement regarding evolution and diversity of organism is correct? Evolution and diversity is correct. Colonization of plant and animal on the land appeared in Paleozoic era. Correct. Colonization of the land took place in the Paleozoic era. Agree. Right? Ordovician time period it happened. Yes. Concentration of carbon dioxide in the atmosphere increase in Archean eon. No, we talk about oxygen revolution. We didn't talk about carbon dioxide revolution. No, that's wrong. Right? Wrong answer. Origin of mammals appeared in Paleozoic. Wrong. Mammals formed during the Mesozoic era. Wrong. In Mesozoic era, origin of human ancestors will appeared. Mesozoic era, origin of human ancestors formed. No. Human formed actually in the Cenozoic era. No. These are all lies. Origin of most present day group of insects appeared in Mesozoic. No. We learn people origin of insects, modern insects, in the Paleozoic, Permian period. So wrong. So we have the answer. Seventh question, answer is number 1. Moving to next page. Moving to next page. Other side. Page. Next page. Question number 8. Which of the following statement? Which of the following statement is correct regarding roots of dicotyledonous plant? Which of the following statement is correct regarding roots of the dicotyledonous plants? Epidermis is multilayered. Cocambium originates from the cortex. Pericycle has meristematic capability. Collenchyma is present inner to the epidermis and distinct pith is present. So here if you take the answers, epidermis is multilayered. Which of the following correct regarding root of dicotyledonous plants? Epidermis is multilayered. No, all plant epidermis is single cell layer. Cocambium originates from the cortex. 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 No, we know people, epidermis is multilayered, wrong. Cocambium originates from the cortex, no. This is about the plant root. Plant root cocambium formed from the pericycle. Wrong statement. Pericycle has meristematic capability, correct. Plant roots, pericycle is meristematic. It is formed in the cocambium, correct. Third one is okay. Collenchyma is present in inner to the epidermis. No collenchyma in the root. Wrong statement. Distinct pith is present. Root, dicot root, distinct pith is not present. It's present in the monocot root. So my answer for question number 8 is number 3. 8 is number 3. Question number 9. Question number 9. Which of the following is incorrect regarding phylum Ascomycota? Phylum Ascomycota. Which of the following is incorrect regarding phylum Ascomycota? Marine or fresh? Marine or freshwater Ascomycota. Correct. Marine members are there. Freshwater members are there. Produce conidia as exospores in asexual reproduction. Produce conidia exospores, correct. Ascomycotes are producing conidia. They are exospores. Agree. Most of these ascomycotes produce sac-like structure called asci. Sac-like structure asci, can agree with that. 8 genetically identical ascospores, wrong. Ascospores are produced by meiosis. So meiosis followed by mitosis. So it can't be genetically identical. They are different. Fourth one is a wrong statement. 9th question, fourth is wrong. Then parasitic or symbiotic members are there. Okay. Question number 10, people select the response which includes organisms that belongs to phylum showing each of the following characteristics in order. Select the response which includes the organisms that belongs to phylum showing each of the following characteristics in order. Segmented body, bare tube feet, radial symmetry, irreversible pharynx, tough cuticle, right? Select the response which include the organisms that belongs to the phylum showing each of the below characteristics. Segmented body, bare tube feet. Now, bare tube feet got to be people accountamate, neither. So, if you take people first one, rugworm segmentation okay, but feather star tube feet okay. Radial symmetry hydra, cedial radial symmetry hydra okay. Then irreversible pharynx hookworm wrong. Hookworm, they don't have irreversible pharynx, neither. Who has irreversible pharynx? Can somebody tell me who has irreversible pharynx? What is the organism with irreversible pharynx? Come on, can somebody tell me? Organism with irreversible pharynx? Who's that organism with irreversible pharynx? Planaria, that is good. Malisha, very good. Malisha. I have some good students in the class. Malisha, yes, very good. So therefore, I don't take first one as the answer. Tough cuticle, maybe mite, ticks and mice, arthropods, it's somewhat okay. Then second one, check that people, centipede, segmented body, centipede. Okay. Bear feed obelia, no. Second one, you can't proceed with that. Just give up that. Third one, millipede, segmented body, okay. Sea urchin, feed, okay. Radius symmetry, coral polyp, yes. Reversible pharynx, planaria. And then tough cuticle, roundworm, okay. Everything perfect for third one. Shall we go for third one, people? Any other alternative answers you have marked? Did you mark anything alternative? Wrong with my pen. Anything else you have marked here? No. Question number 11. Which of the following statement is correct regarding growth process of plants? Which of the following statement is correct regarding growth process of plants? What is your answer? Can you tell me the answer for that? Can you tell me the answer for question number 11, people? Following statement is correct regarding growth process of plants. Primary growth occur due to activity of intercalary meristem. Intercalary meristem, definitely people, primary growth. That's correct. You know, people, intercalary meristem, apical meristems, right? They carry out primary growth. First statement is okay. Secondary growth occurs in all species of gymnosperms and angiosperms. Wrong. All species of angiosperms are not showing secondary growth. Give me an angiosperm which is not showing secondary growth, people. Can somebody tell me an angiosperm which is not showing secondary growth? Can somebody tell me an angiosperm not showing secondary growth? Come on. Angiosperm not showing secondary growth. No, Cecus is not an angiosperm. You can't take that. I'm asking angiosperm showing, not showing secondary growth. Pepper plant. Monocots. Simply you can say monocots. Angiosperm. Right? Palm, coconut, angiosperm, monocots. Right? So secondary growth is in all species of gymnosperms and angiosperm. Wrong. All gymnosperms are showing secondary growth, but all angiosperms don't show secondary growth. Wrong statement. Both primary growth and secondary growth do not occur simultaneously in woody plants. Both primary growth and secondary growth do not occur simultaneously in woody plant. Woody plant, both primary growth and the secondary growth occur simultaneously. Not occur in simultaneously, wrong. It can happen. Now, if you take a mango tree, right, young, the shoot tips, still primary growth occur. And then the lower mature parts, you can see secondary growth occur too. So, they are two events which happen simultaneously, right? Both primary growth and the secondary growth do not occur, wrong. Plant stem increase in length due to lateral meristem, no. Increase in length due to the apical meristem. Increase in girth due to the lateral meristem, wrong statement. Question statement 5, plants show definite growth throughout their lifespan. Plant growth definite, not definite, the indefinite growth. So what you call unlimited growth. They grow throughout the year and years continuously. What you call the perennial plants. Grow like that. Wrong. So the answer must be first one. Any arguments? Question number 11, statement first. Question number 12. Which of the following statement regarding apoplastic pathway is incorrect? They are asking incorrect statement regarding apoplastic pathway. What is your answer for the question? What did you mark, Lamai? Can you tell me the answer that you marked for this question? Question number 12. Which of the following statement regarding apoplast pathway is incorrect? Uptake of soil solution by hydrophilic walls of root hairs provide access to the apoplast. Uptake of soil solution by the hydrophilic walls of the root hairs provide access to the apoplast. Correct. Then people, it consists of plasmodesmata. No. Apoplast does not have plasmodesmata. So why can't we take third answer for 11th question? Third one, both primary growth and secondary growth do not occur simultaneously. They occur simultaneously. Do not occur simultaneously. Wrong, no. Right? Why primary growth occur and in the same time the secondary growth occurred? No. Why plant growth stop the production of leaves? Do plants stop the formation of the growth of the apical meristem when it carry out secondary growth? When the plant is increasing in girth, does it completely stop people the formation of the new buds and the shoots, flowers? Flowers are primary growth. No? Do you see hundreds of flowers are flowering? Right? So if you say so, if the plant stop the primary growth after, right, when it initiate the secondary growth, then none of the plants should produce new leaves. None of the plants should produce any flowers. You see any large number of plants are doing that. Right? You can see every year people, the mango trees are producing new leaves, new shoots. Haven't you observed that? New shoots, so new leaves to produce people, the apical meristem must activate. No. Why? The leaf primordia are produced by the apical meristem. So all these are interrelated, right? According to you, plant is now only increasing the girth and no leaves are produced, no fruits are produced, nothing. Ne? Right? So how can it be correct? Wrongly. Question number 12. Question number 12. Uptake of soil solution by hydrophilic walls of root hairs provide access to correct. To the apoplast correct. It consists of plasmodesmata? No. It doesn't have plasmodesmata. Right? It doesn't have plasmodesmata. Which one? The apoplastic pathway does not have any plasmodesmata. Plasmodesmata present in the symplast. So second statement wrong. Question 12, people, I am taking second one as the answer. Question number 12, my answer is second one. Water and minerals then can diffuse by matrix of cell walls, extracellular space. So first one is wrong. Which one? Which question? Which question? In some species, plasmodesmata may participate. Apoplast, no. Plasmodesmata can't participate. No, wait, I'm in question number 12, right? Question number 13. Water potential reduces in the C2 element. Question number 12, ne? 12, my answer is second. 11th. Which one you say? 11th. I don't know what. Can't you type it fully, people? Your question. 11th. What is this 11th? What's 11th? You want the answer for 11th? 11th, we have taken first one as the answer. 13th, we have taken first one. Huh? Someone says first one. Right. Correct statement regarding phloem loading and phloem translocation is water potential reduces in the C tube element during phloem loading. They are asking incorrect one. Okay, Yahansa, they are asking incorrect one. Water potential reduces when sugars come into the C tube element. What happened to water potential? Does it increase or decrease? Sugars come and collect. What happened to water potential? Can you tell me what happened to water potential? Water potential reduces. Right? Water potential reduces. First statement correct. Second one, most plant phloem loading occurs passively. Phloem loading passive, wrong. Right? Phloem loading passive, wrong. Phloem loading is always active. Phloem unloading is a passive process. So second, 13th question, second is wrong. Specialized companion cells are involved. Specialized companion cells are involved in phloem translocation, correct? What do you call specialized companion cells are there? In some species, plasmodesmata may participate, correct? Phloem loading can occur through plasmodesmata as well. Due to phloem loading, water enters into the seed tube from the adjacent xylem, correct? When sugars enter, water enters also from the nearby adjacent xylem, correct? So my answer is second one for question number 13. Question number 13, second. Right, next one, question number 14. In Caecus, the structure of the ovule which nourishes the developing embryo. In Caecus, the structure of the ovule which nourishes the developing embryo. Wall of the ovary. Wall of ovary. Nucellus. Seed pore. In Caecus, the structure of the ovule which nourishes the developing embryo. Wall of ovary. Nucellus. Seed pore. The structure of the ovule which nourishes the developing embryo. right? The seed pore, male gametophyte, female gametophyte. Now people in cecus, female gametophyte, cecus, wall of ovary. Wall of ovary can't nourish the growing embryo. In cecus, the embryo, this is the embryo of the cecus. Now, the embryo is nourished by the surrounding tissue, right? Can somebody tell me what is this dotted area you call? Can someone tell me what is this dotted area around the cecus embryo called? You learn it very recently. What do you call this? Nucellus is wrong. Nucellus is this part. I'm asking this region. Right? What is this region? Endosperm. Very good. Right? Endosperm. So this is endosperm. This is endosperm. This is the embryo. So endosperm. How endosperm form? Which one transforms into the endosperm, people? What transforms into the endosperm? Very good. Female gametophyte transforms into the endosperm. So my answer is 5 for this question. Question number 14, answer is 5. Why? The cecus, the structure of the ovule which nourish the developing embryo is a female gametophyte. Very good. I have some good students in the class, right? Next one, people. Question number 15. Correct statement regarding opening and closing of stomata. Correct one regarding opening and closing of stomata. Stomata open due to regular expansion of the guard cells in all directions. Stomata open due to regular expansion. Wrong. Neither. Stomatal movement, this guard cell was irregular in expansion. That is why the stoma is going to open. So first one is wrong. Stomata quickly close due to increasing of water potential in the guard cells. No. Due to increasing of the water potential, when as water flow into the guard cell, water potential rises. That open the stoma. Wrong. Energy required for accumulation of potassium into the guard cell obtained from electron transport chain of aerobic respiration. What it says, people? Right? Energy required for accumulation of potassium into the guard cell is given by aerobic respiration. Wrong. Now, who gives energy ATP for potassium accumulation? What you learn in the mechanism, people? What actually helps in potassium accumulation? This is our guard cell. Remember, this is the guard cell. Right? There is unevenly thickened walls present. Here inside a vacuole present. Then this is an important structure. What is that, people? Chloroplast present. So who provides energy? ATP, people, required for the guard cell operation functioning. Now guard cell has to accumulate potassium ions. This potassium ion accumulation need ATP. So who provide ATP, people? Who provides ATP? Chloroplast provide ATP, not by cellular respiration. So energy required for accumulation of potassium is given by aerobic respiration. Wrong. That is given by people photosynthesis, light reaction. Light reaction induces the accumulation of potassium into the guard cell. That is perfect. Correct statement. And then increasing of the carbon dioxide concentration in the substomatal air space opens stomata? No. If carbon dioxide accumulate in the substomatal space, what will happen now, people? Stoma, they are going to close, right? They are not going to open. So question number 15, people, my answer is 4. My answer is 4th. Do you agree or disagree? Any issues, my dear students? මොකද ප්‍රශ්නේ? අට්ටිය අද සද්ද නෑ වැඩි නේද? අද සද්ද නෑ වැඩි නේද? Question number 16, right? Question number 16, we'll do this continuously, right? This is the 21st paper, my dear, right? This is the 21st paper, my dear, right? We can continue this if you can tolerate this, right? And stay continuously on Wednesday night. I'm sure, people, right, you are going to end up your life in medical faculty, right? I'm guaranteeing you that, right? Just stay, right? Put some small effort, and all hundreds of different types of questions we are discussing here, and for you to learn these things properly, accurately, right, to the syllabus, and then definitely, people, right, this cannot be changed. And if you really with interest, if you do it, people, then no one can stop you. And so keep that in mind when you feel tired and sleepy, right? You just right, giving up, right? You are just giving up, right? I don't think it's a good quality that A-level student need to have, right? Don't give up. Just hang on. Life is tough, difficult, right? Difficult means people every time is precious, right? Something difficult, something where most people cannot do means will is something really precious, ne? It's something really precious, something which values, something which values. Cheers. So good things, right? So please do say that to my network provider. No, no. Right. Somehow, I don't think so. Ne? Dialogue telecom, ne? I have sometimes issues goes on. I don't know, right? Sometimes it happened to me also, but I'm not talking about one day. I'm telling you, right? Continuously, hang on, right? Do some good work, right? Cheers. Yes. Do some good work, and then um, no one can stop you, ne? So anyway, people, somehow you do some good work, uh, you are going to the victory. That's all, right? Sir, please continue doing such motivations. Yeah, right. So that's it, ne? So work hard, okay? Every time you are rewarded. Now there isn't anything, right, to give you rewards, right? Nobody. You reward yourself by with your hard work. That's all. No secret behind that. Nothing. Okay. Everything you do for yourself, and you do it for yourself. Eh? When you sleep early, when you eat too much, and then you right, you don't properly study people, then you take right your life. It's your life, ne? You take your life to neither. So unfortunate, right? Work hard every time. You work hard. What's big deal, ne? You work hard. No one can stop you, ne? You have the struggle with yourself. That's all. I mean, you don't have to compete with your friend. No, what to fight? You don't have anything to fight with your friend, ne? You have to fight with your the soul. That's all. Fight with your soul. Fight with your demon and the angel. That's all. Right? Problem is you are having a big demon and a small angel. Wait, the small angel you can't hear. The big demon is talking to you with a loudspeaker. Right? Go and sleep. Eat too much. Ah, nice, good dinner. Ah, yum, yum, yum. Eat and sleep, right? Question number 16. This is the most important period in 2025 students. This is the most important period. You either you get this, you come into the track, or else you lose. Right? Hereafter, right? These couple of months are the determining period. Either you come into the track or else you get lost. If you get lost, people, you can't come back to the track, right? I'm telling you. Question number 16, people, my answer is second. I think it's like straightforward, that theory part, and then check the book. Answer is second. But I don't think you have to check the book, because we discuss nice charts and diagrams and all. Hope these things are still present in your trace of your memory, right? Traces in your memory. Second is the answer. Question number 16, people, select the correct statement made regarding plant growth regulators. Last Monday class, I uploaded that video to the chat and to the website, right? My sleepiness went away after hearing your demon voice. Yeah, you feel me like a demon, eh? Yeah, that's wrong, eh? I'm not the demon, eh? I'm what do you call, right? What do you call me? I'm what do you call me, right? I'm just guardian, eh? No, I'm not an angel. No, I'm not an angel, right? I'm just telling you facts. That's all. Eh? Humble person. Select the correct statement made regarding the plant growth regulators. Plant growth regulators. Right? Plant growth regulators. What is that, people? Correct statement. Ethylene retard leaf abscission. Ethylene abscission. They ask what? Correct statement. Ethylene abscission. Retard abscission. No, ethylene actually promote abscission. Wrong. Gibberellin stimulate pollen. Yeah, definitely. Gibberellin is a sexy hormone. Fruit growth and development, sexual reproduction. Pollen formation, sexual reproduction. Pollen tube growth, sexual reproduction. Everything of gibberellin is like people relate with the sex, right? Nasty hormone, neither. Gibberellin, stimulate pollen growth, correct? Auxin retard apical dominance. Auxin retard, no. Promote, wrong. Abscisic acid promote early seed germination. Abscisic acid, abscisic acid. How many Kurunangal people are here? Please raise your hand up. Lamai, Kurunangal, Lamai, please raise your hand up. Denang jambu me sati eko atau san. 1, 2, 3. Come on, hurry up, hurry up, hurry up. Only 3, huh? 3, 3, 4, 4, 4, 4. Only 4, 5, 5, 5, 5, 6. Only 6, ne? Balagam, balagam. Right, next one, we will select the correct statement regarding plant growth regulators. I'm taking people abscisic acid promote early seed germination. Abscisic acid promote early seed germination. No. Abscisic acid inhibit the seed germination process. No. And then ethylene retard root and root hair formation. Ethylene retard root and root hair formation. Ethylene actually promote root hair formation, especially people we see that the root hair formation, promoted. Ne? So we take second one as the answer. People, my answer for question number 17, second. 17, second. 18, 19, 20, 21, 22, 23. Right, moving to next page. Question number 18, which of the following structural feature of epithelial tissue? Right, structural feature of epithelial tissue is highly vascularized, always consist of several layers of cells, highly vascularized. Structural feature of epithelial tissue, highly vascularized, correct or wrong? Wrong neither. No vascular tissues in epithelial tissue, wrong. And then always consist of several layers of cells, no. Sometimes it can be one single layer, right? And then the consist of the different types of cells, sometimes, but not always, ne? Consist sometimes short cells, tall cells, neither. Not many different types of cells. Inner cellular spaces, intercellular spaces are abundant. No, intercellular spaces. What? Animal tissues, epithelial. No. And then some epithelia, some of the cells do not reach to the epithelium. That's correct. That's correct. Why not? Fifth one is the answer. Yes. Serocertified epithelia, some of the cells are not reaching to the surface. Correct, people. Question number 19. DNA molecule 30,000 base pairs. DNA molecule. Look at this, people. DNA molecule 30,000 base pairs. 30,000 BPS. So how many bases? 60,000 bases. What they say, people? Cytosine is equal to X. Adenine is equal X. Adenine. Select the correct combination. Then thymine is X. Guanine is 2X. So altogether 6X. 6X equal 60,000. Therefore, X is equal 10,000. X is equal 10,000. It has twice the number of purine. Purine is adenine plus guanine is purine. That is, people, 10,000 plus 20,000. Right? That is 30,000. So first statement is wrong. The sum of adenine and thymine is 20. Adenine plus thymine is equal 10,000 plus 10,000. 20,000 correct. Number of pyrimidines is equal to the number of purines. Pyrimidines, adenine plus guanine is 30,000 and Thymine plus cytosine is also 30,000. So C and D, CD, B, D, BC, BC correct. Sum of guanine and cytosine is 10,000 wrong. And then people, the molecular weight of nitrogen bases, adenine is lower wrong. Adenine is higher in molecular weight because it's a purine, two-ring structure. So BC must be the answer. Question number 20. And question number 20. Correct regarding leaves of siamase. What is siamase? C4 plant. Siamase is C4 plant. Well-developed grana. Well-developed grana. Well-developed grana found in mesophyll cells. Well-developed grana present in mesophyll cells. Mesophyll cells, well-developed grana, correct. Then chloroplast, well-developed grana in mesophyll cells. Poorly developed grana present in the bundle sheath cells, correct. Chloroplast of mesophyll cells have abundant PEP carboxylase enzyme. Chloroplast of mesophyll cells have abundant PEP carboxylase enzyme. Chloroplast of mesophyll cells have PEP carboxylase enzyme. Abundant, correct. Chloroplast of mesophyll cells. Wrong, neither. Not chloroplast, napule. Where do you find that? Cytoplasm. So this is given as chloroplast. So A correct, B wrong. Bundle sheath cells produce pyruvate in presence of light. Bundle sheath cells produce pyruvate, correct, neither. Bundle sheath cells convert malate to pyruvate, correct statement. You learn that happen in C4 pathway. Right? So C correct. D, bundle sheath cells contain rubisco abundantly. Bundle sheath has rubisco, correct. So D correct. Liberation of oxygen take place mainly in bundle sheath cells. Liberation of oxygen take place mainly in bundle sheath cells. No, oxygen is liberated in people mesophyll cells. Wrong. So I'm taking ACD, second one. ACD, second. Agree? Adlama ACD agree, ne? The true statement out of the following regarding determination of the rate of photosynthesis using Audas microbiorate. Now, you know Audas microbiorate? Do you know Audas microbiorate apparatus? Audas apparatus lamai? Audas microbiorate. Audas microbiorate is designed to collect people oxygen released by an aquatic plant. There is a capillary tubing to measure the volume, and then people, there is a syringe over here to pull this volume. So inside is soap water filled, and then you are inserting the chute, cut chute, right? Turn upside down like that, and there is a test tube filled with water, and then outer water bath present like this. This water bath. Light source is kept near. This is a light source. This is a light source. By adjusting the light source, we are now changing the light intensity. This outer water bath is now maintaining people the required temperature for the experiment. Plant is now cut chute is turned upside down, and this is upright. The upright stem piece present. This is for uniform release of oxygen occur from the cut surface. Oxygen go and collect here and then forms a water column, the air column, and by pulling the piston back, you can draw this into this area so that you can easily measure the volume against this calibrated tube. This is how you do it, right? This is how you do it. This is Audas microbiorate apparatus. Why you use this, people? To measure oxygen volume release in photosynthesis. A sodium bicarbonate solution is added to the test tube to prevent oxygen being a limiting factor. Sodium bicarbonate solution is added for oxygen. No. Sodium bicarbonate is added, people, for carbon dioxide to be available throughout the experiment. First statement is wrong. The setup is kept in water heater to keep the temperature constant. No, we don't keep it in a water heater. Water bath, water heater, wrong statement. The hydrilla plant is kept upright to make sure the air bubbles collect uniformly, correct. So that they won't trap any under the leaf and all, so it will uniformly release from the cut surface, correct. Water is aerated before the experiment to oxidize organic matter. No, no, maybe to provide carbon dioxide, we don't have to oxidize the organic matter. No, we don't take such polluted water, we just take clean water, people. The table lamp is used to determine the variation of the rate of photosynthesis with light intensity, correct. You bring it closer, you increase the light intensity. You take it away, people, you decrease the light intensity. So, I am taking C and E as the answer. Question number 21. C and E කියන්නේ 5. 20, what is the answer? Will I have taken A, C, D for 20. I think I'm A, C, D 20 here. Make එතකොට ළමයි A, C, D කියන්නේ number 2. 19th question, people, I took B and C. B and C. හරි ඒ කියන්නේ 5 once again. B and C. Sorry about that. Error in marking. Question number 22, correct comparisons regarding specific character, phylum, flame bulb, platyhelminthes. What do you say? Correct, ne? And then people, metameric segmentation, nematoda. No, they don't have any segments, no. Joint appendages, arthropoda, yes. Tube feet, echondamata, correct. Cilia caudata, but it's not characteristic, ne? Wrong. So they ask people a specific character, ne? So I'm taking ACD. 22 A, C, D means second one. Cilia present, but you can't take it as characteristic, right, people? Question number 23, which of the following features are common to all phyla of seed plants? Pollen grains. No, not present in all phyla. Wrong. Double fertilization. Common to all phyla. Oh, they are asking seed plants. Oops. Seed plants, huh? Pollen grains present in all seed plants. Correct. Gymnosperm, angiosperms. Yes. Double fertilization. No. Non-motile sperms, no. Some secus, motile, no. Ovules, yes. Xylem vessel elements, no. A to all phyla. A and D. A and D. A, D. 5, ne? A and D. 5. Common to all seed plants. A and D. 5. So 20th question, why the statement B is incorrect? So why E wrong? Xylem vessel elements present in all seed plants. Wrongly. Xylem vessel elements are not present in secus. Do they have? Xylem vessel elements in secus plant? Netophyta xylem vessel elements are there. But you don't see xylem vessel elements present in secus. Gymnosperm? Wrongly. Right? Ovules present. Ovules are there. But ovary wrong. So in 20th question, in 20, why the statement B is incorrect? Why statement B is incorrect? Chloroplast of mesophyll cells have abundant. Chloroplast of mesophyll cells, no, not in the chloroplast. Ne? The PEP is available in the cytoplasm. I told you that, ne? Present in cytoplasm. All right. Okay. Right, moving to next page, people. Last page. We have a graph to work out. Did you work out the graph? Lamai, did you work out the graph? I have given you some EC data sets. Right. Try to work it out and then draw the graph. Can you tell me these values you have obtained? 15.15 m. What is the value? 0.2 minus 0.2. You tell me the values now. 0.20 m. What is the obtained value? 0.20. Can you tell me the value? Come on. Come on, tell me the value. Right. Solo potential. Come on, students. What's wrong with you all? You didn't do this? You didn't do? Question number 1, you didn't do that? You didn't do? I'm going to check, huh? Right. Here, bring this paper to the weekend class. Did you answer the structure they say yes or no? No. No. Why not? So there is a table given. There is a table given. So how do you work it out? Initial curvature, final curvature. So percentage change in curvature. What is percentage change in curvature? What is percentage change in curvature? Final minus initial. Final minus initial. Final minus initial. Right? Divide by initial. Ne? Divide by initial. So what is this final? 88 minus 110. Divide by 110. Divide by 110. So what do you have here? 88, 1200, 22. Ne? 22 divided by 110. 22 divided by 110 means here 22 divided by 110. This is 11 divided by 55. 11 divided by 55. Second 1 and 5. 0.2. 0.2, right? 0.2. Right. So for 0.15, I'll erase this part now. 0.15 m, 0.2 minus is minus ne? Minus 22, right? So minus 2. Minus 2. Right. We'll do for the second one. What is that? Final is 97 minus 108 divided by 108. What's the difference? What's the difference? I just work out this data set. Maybe not sure whether I did it with enough attention. We'll see. 108, 97 minus 108 can equal high. I think I'm wrong. Divide by 100. And we get some small decimal values. 0.1 with Rogeno. Yes, 0.1 with Rogeno. Okay, I think I'm not given the data set properly, but let's try to work it out. 0.1 minus 0.1. So here 0.20 we get minus 0.1. Next one. Next one. What is that? Next one, people, you can take 106. Final is 106 minus 105 divided by 105. What is that value? Plus 1, ne? 1 divided by 105. Okay, almost 0, ne? This is 0, almost 0. 0, right? 0. Then, so 0.25 m, 0. The next one, people. Next one. How to take next one? Final curvature 115 minus 104 divide by 104. Initially 104. So 115 minus 104 divide by 104. Kila kiyanne plus point plus 0.1. So plus 0.30 m plus 0.1 plus sorry the. There was a 35 letter. 35. 35. 128 minus 6 divide by 106. Kidana value beka? 128 minus 106 divide by 106 lu 0.20 plus 0.20. So 0.35 m plus 0.20. Looks okay in the data set. I think it's quite okay. Yeah. So should the answer be multiplied by 100 to get the percentage change? Is it, yeah, okay, ne? Right? Percentage. Initial into 100. Yeah, okay, fine. Right? Into 100. Huh? So then you can take here. This value then minus 20%. Ne? Minus 10%. If you do that, this is 0. This is 10% plus 20%. Doesn't matter, eh? Plus plus. Now you can draw the graph. Okay, you can draw the graph, ne? But thing is now we have the isotonic solution 0.25, ne? Definitely isotonic solution is 0.25. You can draw the graph now. You can draw the graph. You can draw the graph. You can mark it like this: 0.15, 0.20, 0.25, 0.30, 0.35. This side m. Right. This side we are taking percentage curvature. This is given percentage curvature. Some error, ne? So it should come with people percentage 100%. So small issues are there. Here people will take that 0, 10, 20 plus plus 10, 20 plus this side minus 10 minus 20. Yeah. So then 0.15 people you take minus 20 here. This is minus 10. This is almost here. This is plus 10 and this one. So you get a straight line, like that. You get a straight line. So what they ask now? Complete the above table. Draw the shape of the alocasia leaf stalk strip after immersing in 0.2 m sucrose solution. 0.2 m sucrose solution. Then people, this is soon after slicing. This is the peel side, ne? This is the peel side. This is soon after slicing. 0.2 m means, well, water is going to enter. Water is going to enter. Then it's going to curve further like this. So if you draw it like this, better. This side now towards the peel side, it curve more because of this entry of water. Right? So you draw it like this. Because we are immersing in 0.2 m. This side, 0.3 m and all people, we can see that they are hypertonic. So it's going to curve to the other side. All right. So then calculate the water potential of the alocasia leaf stalk strip based on the graph. Water potential now we learn people 0.2 m is 0. Right. So we'll take that. This my graph is not good. Right. This is the 0.2, right? 0.25. So what are you going to take? 0.25 is what is the solute potential? Minus 680. So water potential is minus 680. Right? Alocasia leaf stock, calculate the water potential minus 680. Got it? Right? Because this is the isotonic solution. This is 0.25. Yeah, 0.25 according to the table. 0.25 is, make a psi s value. Psi s of the solution is minus 680 kilopascals. Right? You know, people, this psi s of the solution is equal to water potential of the cell tissue is equal minus 680 kilopascals. Minus 680 kilopascals, right? I think you can work it out. Find the pressure potential of alocasia tissue immersed in 0.3 m solution. They are asking 0.3 m solution pressure potential, right? Now you have immersed the tissue. Find the pressure potential of alocasia tissue immersed in 0.3 m solution. We can't find that. Can you cut that question, people? No, we can't find that. We can't find it because we know water potential. We know water potential of the tissue we know now. Solute potential we don't know. We don't have any clue regarding solute potential. This can be done, people, only for rho u. Right? Can you write there, people? This cannot be calculated, right? This can be done only for rho u. You can't do this for this tissue, right? Right? This is completely wrong question, right? Put a note, right? You can find this only for rho u, not for alocasia, right? Because we had to make that assumption, 50% plasmolysis, incipient plasmolysis, that thing is coming under the rho u, not for this. Wrong statement, right? Then people briefly describe the role of photoreceptors. Let's check that part. Let's check that question. This is our last work, right? So check the scheme for the question. Let me give you the marking scheme for that question, people. This one, right? Oh no, that's for the juniors one. I had 2 classes today, right? Juniors, this is your one. Right? This is your one. Right, describe the role of photoreceptors. Describe the role of photoreceptors in photomorphogenesis. You learned this theory part recently. Photoreceptors in photomorphogenesis. Right, so this is my answer. The 2 major types of photoreceptors. Photoreceptors in photomorphogenesis, right? 2 types of photoreceptors are there, blue light photoreceptors present and phytochromes are there. You learn, people, blue light photoreceptors are 2 types. What are those 2 types, people? The phytochrome and present, sorry, cryptochrome is available, phototrophin present, right? Then you learn, people, the phototrophin is involving in operation of stomata, cryptochrome is involving in inhibition of hypocotyle elongation, and cryptochrome is also important, people, for what? Cryptochrome is important for the phototropism. Then you have to talk about the photoreceptors, phytochrome photoreceptors. It exists in 2 forms, people. What are those 2 forms? They exist in PR and PFR form. We learn the details, people. This is helping in plant spacing, seed germination, and also, people, this is involved in controlling flowering, right? So this description you must write, describe the role of photoreceptors, you must write under this section. Then second part of the question, people, I'm asking you, state the function of plant growth regulators, auxin, gibberellin, and abscisic acid in responding to different stimuli, right? Auxin function, gibberellin function, and abscisic acid in response to different stimuli, right? So therefore, you must write regarding, people, these 3 hormones. There is a description given, so you better write that part. Please take, people, a screenshot. Take a screenshot. Check this answer. Hope you have written this, right? Hope you have written this answer. Haridlamai took the screenshot. Haridha, Haridha, Haridha. I'm closing, right? So that's Right. Okay, that's the end of our today's discussion, people. Then thank you very much for your participation, and I'll be meeting you people soon in our normal regular class. See you.